Hello, sir. Hello, anyone? Hello,
Yes, sir.
slide
Yes,
is visible.
Yes, sir.
Okay, I
yes,
will take it. There was some internet problem, so that's why we could not start well in time. Uh, Application of reproductive and biotechnological tools for genetic improvement of livestock and poultry. The handout I will send you in your WhatsApp group. Okay. Now here, biological tools. That is the different techniques developed in the field of biotechnology. So with this system, so many techniques are there. One by one, we'll discuss. So these techniques can be utilized for genetic improvement of our livestock and the poultry species. So then different techniques which were developed were in the field of reproduction genetics and the related aspects. So what we do, these different techniques of the biotechnology, they work through four factors. That is, they increase the genetic variations, because through the genetic variations only we can do some selection in the breeding program. Then they increased or they help in the accurate evaluation of the breeding value, that is accuracy of selection. Then they reduce the generation interval, which is very high in case of progeny testing. So that performance evaluation and all those things. Then it implemented in case of livestock animals. Then another one is multiple ovulation and the embryo transfer. So I will give you then another one, embryo production and anything. Using techniques, we can increase the production performance of our livestock animals. So here, then coming to this artificial insemination, it was in case of cattle, and then what we can do is using a very few superior males, we can cover so many animals. So in that, there is a drastic improvement in the process of breeding. Then this with the technique of artificial inseminations, we can go for progeny testing, where we a very good number of females to be in from a particular bull. But a bull can be evaluated on the basis of progeny, so that way it is the accuracy of selection. Among multiple ovulation and embryo transfer is MOET, which is mainly used in case of nuclear utilizing MOET. Here what we do is we increase the number of offspring, especially from their female, because female they do produce offspring in their lifetime. So by using these multiple populations from a particular superior female, we can have more number of offspring. Then here the offspring produced from such type of multiple ovulations, they are evaluated on the basis of their half or full sisters performance, just like that in open nucleus breeding scheme. So it helps in the reduction of the generation interval, time required is reduced. Then another merit of this technique is it has in the repeat multiplication of superior phenotype, especially the female. Then oocyte harvesting and oocyte maturation and in vitro fertilization, that is IVF. So here we, by the use of multiple ovulation and embryo transfer here, we can get hardly 20 or less. The IVM and the IVF, it is a factor of 5, means multiple times. Even use in case of pregnant animals also, as well as in those heifer, that is pre-pubic animals. So in this way, it increases the selection of male and female. Here transfer or embryo cloning. Here what we do is transfer of totipotent nuclei. Totipotent nuclei means the nucleus contains the genetic material and to grow as a full individual. So in this way, we can have any identical twins or cloning. So what we do is, as you know, Dolly sheep was cloned, and by using somatic cells, that is body cells, it helps in the cloning of a proven outstanding animal. If we identify male or female outstanding animal in a population, that can be cloned by using these cloning techniques, and it is on the means for conservation of endangered breeds or species. So here's some photo, this one. Flows are how we do this cloning here. Embryo transfer. Here is one donor embryo. Then recipient embryo, we have removed the nucleus of the recipient oocyte. Then this donor embryo, the nucleus, we have taken it out. Then this is inserted into the enucleate. That means the nucleus which the cell for which the nucleus was removed. There we have transferred this nucleus. Then by electrofusion, they are fused together. Then this cell with the new genetic material will behave as a normal embryo. This can be transferred to a recipient individual. So now coming to the embryo cryopreservation, sperm, spermatozoa, and all that can be a period of time through cryopreservation. Just like that, embryo also we can collect, and they can be kept or stored using this liquid nitrogen for future use. Now, embryo splitting. That means once one embryo is collected, it can be divided into many embryos. One embryo offspring. This particular embryo is from some outstanding animal. So in that way, so many of us superior offspring we can have from one particular individual by doing this embryo. So it can use for environment here is, say for example, in case of genotype environment, that clone embryos or embryos where they have the same genetic materials, is different of individual, they can be rare in different types of environment, so they perform in different agroclimatic conditions. And at the same time, for selection of carcass traits, some of the items may be sacrificed to evaluate their carcass characteristics. The remaining live clones or individuals or identical twins used for purpose. Then sex selection here. With the development of the pro-cytometric sorting, we can sort these the gametes which are having X or gametes. So this might be one of the method for more number of F particles. Then embryo extraction by microbiopsy, determine using Y-specific sequences. So these are somewhat your PG level, but at least you try to remember that using molecular techniques also we can identify a bearing chromosome, Y chromosome. In that way, we can have that particular individual of our interest. Then coming to the genetic biotechnologies here, it helps in the genome analysis, and then it helps in the manipulation of genome. Here, let us see one by one. Genome analysis here, by this method, we can molecular or DNA markers, and molecular or DNA marker is example means what is your identification? Is there anything? Yes, there is a mole, there is a scar mark, and all those things. Those are the marker. So here's a DNA segment. It is a small segment of DNA which is associated with and hence segregates in a predictable pattern as the trait. Means we have some economic traits. If a particular segment of DNA is associated or linked together with those genes responsible for a particular trait, then that DNA segment we can identify whether that any of the individuals are having that DNA segment of our interest. If that DNA segment is present in that particular individual, we assume that the individual is having the genes responsible for it. So then genetic marker, then a genetic marker need to have an of the example marker. Say for example, double muscling gene in Belgian blue cattle, then pole gene, then Weaver syndrome in brown Swiss cattle. So these are the some of the markers identified for. Then these genome analysis are what we need. Then how to identify those particular DNA segment which can be used as a marker. This technique needs the screening the genome or genes for large effect, large Mendelian genetics. It is similar to your dominant gene. So in case of quantitative traits is or economic traits, they are by pairs of genes. They are known as polygenic traits. So there we don't use the word dominant and the recessive. We use the word large effect, means out of those many pairs of genes, some gene may be having large effect. So like this, then by the techniques of statistics and organ analysis, we can analyze by using this linkage analysis. Linkage analysis means if the two genes are located on the same pair of chromosome, they have a tendency to go together from generation to generation. In that way, we can identify a particular DNA segment which is linked with some of the economic traits. Then the DNA markers, so far I am giving here some of the names which are commonly used as a DNA marker. So RFLP, that is full form is restricted fragment length polymorphism, mini satellite, microsatellite, that is randomly amplified polymorphic DNA, AFLP, amplified fragment length polymorphism, DNA fingerprinting, APPCR means arbitrary primer. So here, what actually AR is not that much required in the UG level. What you should know is, yes, so the DNA markers, you should remember all of them or some of them. Okay. Then, what are the applications of these molecular markers? Some of the applications are identification of parents. We can identify the parents. So, for example, some disputed offspring is there. Which one was the parent? We are unable to have the idea. Then by using the molecular marker, we can identify actual parents of one individual. Then estimation of genetic similarities or dissimilarities. Here, so many individuals are there in a population, we can estimate what are the genetic similarities. So in case of our statistical methods, we use this one relationship coefficient. What is your relationship with your brother, with your father? 50%, 25%, like that. So by using these molecular techniques or molecular markers, we can identify those similarities between two individuals as well as the distance or dissimilarities. Then what is MAS here? Marker assisted selection. I will give more information in the down slides. Then it can be used, this sex determination molecular marker as in carrying gamete, carrying gamete like that. Then identification of carrier animal for any deleterious or harmful gene. So we can identify whether this particular individual is having any harmful gene or carrier so that it can be removed from the breeding program. So like that, this molecular marker can be used in many fields. And here is one marker assisted selection. For your short notes, it is important. What actually it is, let us discuss in brief. Otherwise, it is a very big topic. Yeah, I said already that selection of a trait is our interest based on a genetic or molecular marker is called marker assisted selection. See, by screening the individuals present in the whole population, so there are 100 individuals, we have screened, genome analysis is done. by using suitable techniques. Then we identify some of the individuals which are having the DNA segment of our interest. So if those individuals facing that DNA segment of our interest, they are selected and used in breeding. So here, identification of superior individuals is based on the presence or absence of DNA marker. So such type of selection is known as marker assisted selection. So therefore, it is a type of indirect selection because we are not evaluating the individuals on the basis of the observed economic traits. We are selecting the superior individual in the indirect way by targeting the DNA segment, presence or absence. Then it depends on the identification of association between the genetic markers and the link QTL. So you should know what is the form of QTL. QTL means quantitative trait loci. So these things are required for marker assisted selection. Then, as you know, individual selection is very effective for high heritable traits, but for low heritable traits, individual selection, that is selection based on the phenotypic performance, that is not effective. And then we go for progeny testing. Then again in progeny testing, one drawback is it takes a very long time, that is generation intervaries, and it is very costly and all. Then here, marker assisted selection can be used for low heritable traits just by identifications of those desirable DNA segment, their presence or absence. Then marker assisted selection can also be combined with phenotypic information, which will increase the efficiency of selection or accuracy of selection by 10 to 20% over. Then this marker assisted selection, as you know, DNA, once it is decided at the time of fertilization, it cannot be modified or it cannot be changed. So presence of a particular DNA segment of our interest can be done at the very early age of an individual. So it reduces the generation interval. Means selection can be done at a very early age. Then it has a very wide scope for selection of the disease resistance. So it is very difficult to go for our traditional type of breeding system, especially for disease resistance. So there we can utilize this knowledge of the molecular markers, that is marker assisted selections. So far in the different parts of the world, we have there are so many projects. Then one and two I am mentioning here: tolerance to African trypanosomiasis in Endama cattle. Emma is a cattle breed. They are known for their resistance or tolerance against the trypanosomiasis. So that study is also going on. Then another one is the resistance to endoparasites in a red Maasai sheep. So these are some of the markers, assisted selection. People are working on this aspect. Then coming to the transgenic animals here, you may be having some idea what actually these transgenic animals, the genetic or genotype of an individual can be altered techniques of transgenesis. That is a process of introduction of a gene or a DNA segment of our interest to some other individuals. So the individual which receive the genetic material from some other source that is known as foreign DNA, those individuals are known as transgenic animal. And this technique was first used in case of mice, transgenic mice, that was by Gordon and Rudel in the year 1992. Afterwards, in the other species like rats, rabbits, sheep, etc. Then, successful production of the majority of the studies for transgenic animals were carried out in case of pig. Then, ability to insert new genes for economically important characteristics like fecundity, resistance or tolerance to environmental stress. So in this particular field, it will give a major plus in the genetic improvement program of our livestock animals. Then you might have heard this one, transgenic technology or transgenic to produce some of our medically important proteins such as insulin and the clotting factors. Clotting factors those genes, this one they have cloned in case of sheep milk of the sheep, where these clotting factors, that is the human factor 9, they are released, their proteins are released in the milk of the sheep. So those sheep are known as transgenic sheep. So accordingly, we modify their genetic material so as to harvest the important proteins useful for our medical field. Then growth hormone encoding gene constructs have been transferred to sheep also. In this way, we want to know whether the sheep which receive the growth hormone gene, that is the transgenic gene, transgenic sheep containing growth hormone, whether they are having superior growth or not. Then transgenic sheep also was produced for expression of the Vishna virus enveloped gene. So in this way means science is now developing like anything, especially in the field of biotechnology. So here I am finishing in this one. Then now another one, I will try to take the just wait. So now next topic is breeding strategies for the improvement of dairy and the dairy cattle and the buffalo. There was many of the techniques. See, cattle and the buffalo genetic resources in the country, as you know, the number of recognized cattle breeds in our country is 50. Then for buffalo, it is 17. That is 50 and 17. So you should know for your fill in the blanks or multiple choice also. So we have a very good number of animals. However, around 80%, that is only 20% we have evaluated or characterized, but the remaining 80% still they are considered as nondescript types. That means so many cattle breeds will be there in the days to come, means in the future. People are doing this characterization at the phenotypic level as well as at a molecular level. So these different types of recognized cattle breeds as well as nondescript cattle, they belong to different agroclimatic conditions. So we cannot have a similar type of breeding plan for these different breeds. Now, as we know, most of our indigenous cattle and the buffalo, they are known for their poor production potential. Then we don't have a very suitable breeding policy. Most of the time we are facing this, there is improper breeding policy, or there is no breeding policy for this particular type of population. Then we have a very little supply of or sufficient supply of this nutrition, health coverage, and the management. So in this aspect also, people are always claiming that we don't have this adequate nutrition, we don't have this proper health coverage, we don't know this proper management and all those things. So these are the many factors which are responsible for poor production potential of our indigenous cattle and the buffalo. So what is a breeding plan? So it is the proper combination of selective breeding, outcrossing, grading up, crossbreeding, like that. So in this way, we have to combine selection. You identify parts, the superior animals, males and females, we have identified them in which way we should go their breeding. So in this way, that combination is known as a breeding plan. Now, the formulation of a breeding plan, it involves what are the factors involved in the formulation of a suitable breeding plan. So what is in our mind, what we want to improve economically in this particular cattle? It may be for milk, it may be for draft purpose, like this. Then which breed as a foundation stock, whether this existing breed is sufficient or we need to introduce some individual, some superior individual from outside. Then whether the genetic improvement is possible within the herd or population, or else we need to introduce some superior genes from outside, right? Outside means, say your farm is there, the animals, their production performance is very low, then you need to introduce some of the superior genes or animals in the form of animal genes will be introduced in the farm. So those outside genes or animals may be of the same type of breed. If you want to maintain Thar Parker cattle, then you want to introduce superior Thar Parker cattle from some other source, or else if you are interested to mix your breed with some other breeds, that is known as cross breeding. So here, now you please keep in your mind, those are the background informations. The breeding strategies for cattle may be of the entire country. Grading up, as you know, grading up, this term is used for nondescript animal. So the improver breed, that the recognized breed, may be of our zebu breeds. Then grading up or crossbreeding of the nondescript cattle with exotic breeds means so many nondescript cattle are available in our country. Many states they are having. So some of the states they may prefer for grading up using Zebu cattle. That is our Sahiwal, our Thar Parker, our Red Sindhi, our Gir, or else they may go for exotic cattle like Holstein Friesian or Jersey. Then we also have some well-defined animals like Sahiwal, Red Sindhi, Gir, Kankares, Thar Parker, Ongol, and all like that. So for those breeds, we don't like to mix with other breeds. Means we prefer to maintain their purity. So in that situation, what we do? Selective breeding. Then what actually is selective breeding? Let us discuss one by one. So now, for grading up of our nondescript cattle using our own cattle breeds like, so the recommended breeds may be Sahiwal, as I already mentioned, Red Sindhi, Gir, Thar Parker, or else it may be Haryana, it may be Ongol, or it may be Kankres. These are known as dual purpose breed. So as per the requirement of a particular climatic zone, we should select or recommend whether the mixed breed of our country or the dual purpose breed should be recommended. So then for our Northeast region, we recommended Red Sindhi, Sahiwal, Haryana. So these are the recommended well-established breeds of the country for use in the grading of the nondescript cattle available in the Northeast region. So here you are very much familiar with this breed, but I am telling this first one is Sahiwal. Then next one, some sports, white spots, they are there. This is the Gir. Then next is Kankres, very long hair. Then another, the last one is the Red Sindhi. So these are the well-defined breeds of the country. Then grading up or crossbreeding of the nondescript cattle with exotic cattle. Here, what we will do, which of the exotic cattle, which one should be used. So in that situation, we have to identify for that particular agroclimatic zones, what is the position of the availability of feed and fodder resources. Then what is the situation about the veterinary health facilities or marketing facilities. Means as per the market demand, we have to go for the type of the exotic breed. So then here one by one I am telling, Holstein Friesian commonly use exotic breed for upgradation or crossbreeding of the native cattle. So this Holstein Friesian is recommended for those areas where irrigation facilities is there. Due to this, there is adequate feed and fodder supply, and also veterinary health services. As you know, the crossbred offspring produced from Holstein Friesian, they are very prone to many of the diseases, especially parasitic diseases and all. So in that situation, we need a very good facilities of the veterinary health services, as well as, since they are a very huge animal, big size animal, they need a proper supply of feed and fodder only, then they can perform. Then Jersey, especially since they are small in size and known for their adaptability, that is feed conversion efficiency and all. Jersey is the exotic breed recommended for Northeast region as well as coastal areas like Kerala, Goa and all. Then brown Swiss is especially recommended for Kerala state. And by using this brown Swiss, Kerala Livestock Development Board, LDDB, they have evolve a new breed known as Sunandini by using brown Swiss and their local cattle. So here is the top. This one is your Jersey. Then white and black spot is Holstein Friesian. Down is brown Swiss. Then, if we are doing this level crossbreeding and all, then what should be the exotic inheritance? So we suggested that it should be in between 50 to 75% or average of 62.5%. Then, in which area this 50% level should be used? That is in the area where these veterinary health services and the supply of the good feed and fodder, if that is poor, so in that area, we should go up to only 50% so that the animal can survive in this scarce condition of scarcity of food and other facilities. So that is remote area. This area comes in the remote village and the remote areas. Then 75% up to 75% level of the exotic inheritance, we can go in the area that is urban and the city areas where adequate facilities in terms of feed, fodder, and veterinary health services and marketing facilities are available. Then selective breeding within the Zebu cattle. We don't like to lose our Zebu cattle. I mean, some of the well-defined, well-recognized breeds are there, like Sahiwal, Red Sindhi, Gir, Cancres, Ongol, Harparker, like that. So for this one, what we can do is the suggested breeding plan is selective breeding. That is what? You select the best males and the females and breed them together to maintain the purity of the breed. Then this selective breeding, it can be done by, generally selection is done in case of males. So by using associated progeny testing program, so here is one word missing, associated progeny testing program, that is identification of the superior males, or nucleus breeding system with moed or without moed. So by following these two techniques, you can identify the superior male so as to use for breeding purposes. So in the exam, when these, if it comes in the exam, what will be the breeding policy for local cattle of Mizoram, then you can write it, but you should also try to write something about what is associated progeny testing program, then what is ONBS in a very brief note. Then associated progeny testing program, as we know, it is used to increase the size of the population. So by incorporating what? Organized farms, our college farms, government farms, military dairy farms, gausalas, farmers, animal. Then ONBS with or without moed can also be used for identifying the superior male. Then in case of buffaloes, as we know, we have the world-class buffalo breeds in our country. So the two breeding strategies recommended for the improvement of buffaloes in our country, they are grading up for nondescript buffaloes and selective breeding for well-defined breeds. Then this grading up, for doing this grading up, the improver breeds to be used are Mura, Nilirabi, Mahesana, Surti breeds, and I'm showing here the photograph. First one is Mura, next is Nilirabi. Nilirabi means white spots at the forehead, legs, tail, switch of the tail. That is there. That is known as Nilirabi from Punjab. Then down is the very huge horn. That is your Jafrabadi. Then another one, the last one is your Mahesana. So after 4 to 5 generations of upgrading, as we discussed already in the upgrading, that your nondescript animal will be almost converted into improver breeds. Then for selective breeding, again here for selection of the superior breeds, as in case of cattle, we can go for associated progeny testing and nucleus breeding system without moed or with moed for identification of the superior male. So here also you have to elaborate up to some extent when you are writing in the exam. So this is the end of this one and today's class. Okay. Thank you all. For all of you, I am finishing your class here. Okay, bye.
Thank you, sir.
Okay, thank you. Welcome.